All right, well, we're ready to go. Thank you very much for sticking with us. And now we're going to open up our translational panel. And I would like to introduce the leader of that panel, Dr. Jong, who is a professor of neurosciences and pediatrics at the University of California, San Diego. He's also currently the division chief of pediatric neurology at the Rady Children's Hospital in San Diego. Dr. Rowe's clinical expertise is in general pediatric neurology and pediatric epilepsy, with a special emphasis on pharmacological therapies and metabolic approaches toward epilepsy treatment, such as the ketogenic diet. His main research interests are the mechanisms underlying the anti-seizure and neuroprotective effects of the ketogenic diet and its clinical variants. Dr. Rowe.
Thanks very much, Adam, for that introduction, and thank you all for coming back for the second panel session entitled Translational Panel. This is a shorter panel. I don't know if that reflects kind of the state of the art with regard to translational knowledge, both from the basic as well as the clinical side, or whether that is sort of a reflection also of the continuing translational divide that exists between the basic science arena and the clinical world. But I hope that within this brief session, we'll be able to pose some of the more important questions that we need to tackle in order to make further progress in the field of metabolism-based treatments for epilepsy and perhaps other neurological disorders. Before I introduce the panel members and to begin the discussion, I wanted to have a few comments about sort of the concept of translation and what we'll try to focus on. Next. So what is translational medicine? It's defined somewhat differently depending on who you talk to. And it's often, this is one sort of definition provided by Science Translational Medicine. Quotes, often described as an effort to carry scientific knowledge from bench to bedside. Translational medicine builds on basic research advances, studies of biological processes using cell cultures, for example, or animal models, and uses them to develop new therapies or medical procedures. But, you know, although historically we've always thought of this as bench to bedside and more recently bench to bedside and back, realistically, it should really be a bidirectional sort of perspective. Next. So the idea that clinical insights and knowledge or the questions that arise in clinical research should help guide what we find in terms of basic mechanisms, the basic translational implications. It really should be bidirectional, and I think this is really the framework that we'd like to pursue for the future. Next. The other topic, which has been sort of the holy grail, or one of the holy grails of not just the field of metabolism-based treatments, but perhaps all of medicine, is this concept that we need to find some way of quantifying or measuring something that reflects the disease process, a response to therapeutic intervention, something that gives us some objective measure of what's really happening to the patient and with respect to therapeutics. The best recent definition of biomarker is one that was framed within the recent FDA NIH biomarker working group, the so-called best biomarkers, endpoints, and other tools resource that was recently published. Defined characteristic that is measured as an indicator of normal biological processes, pathogenic processes, or responses to an exposure or intervention. Now, while that may seem very direct and straightforward, it's deceptively simple and very, very hard to do, especially for complex organs such as the brain and for complex diseases such as epilepsy. Next. The question that many of you have asked over the years is there a biomarker related to the ketogenic diet or any metabolic treatment? With respect to metabolism-based treatments for epilepsy, despite efforts by multiple groups throughout the world, at present, we do not have any very validated biomarkers, either direct biomarkers or surrogate biomarkers, either in humans or in animal models. And this, I think, is one of the key questions and challenges that we face as investigators on all sides, is to try to develop one or more of these to help guide research, both clinically and experimentally, but also more practically in the realm of ketogenic and metabolic treatments for patients with epilepsy. Next. So one of the other questions we'd like to tackle here with our small panel is sort of an obvious one. We study many of the basic translational questions and issues in animal models or in vitro preparations, but how well do they really reflect what happens in humans, right? So the idea that, should we focus further on animal model research? What kind of benefits would there be as a result of that? Or should we be investing more in so-called human biology, where human studies applying new technologies that give us insights into the brain? Big data approaches, imaging approaches, things that we have only recently been able to access within the last 10 to 15 years. So this is a question I think that will be a fundamental one to address. Next. So in terms of that comparative anatomy and neurobiology, we often think of, the human brain in the context of other, species. And since most of us actually focus in the bench scientific arena in rodents, rats and mice, it's often said, well, the human brain is not the same as a rodent brain. And it's not just a matter of size. And does size really matter? In fact, the human brain is really not the largest brain that's out there. The African elephant, for example, has a much larger brain in terms of mass than we do as humans. So in this figure here, you can see the comparative anatomical differences, at least grossly speaking, with various species. Next. And while we think of this as sort of the first approximation, when we drill down to the various hierarchical levels of inquiry and study this, we find fundamental differences. And this is another theme that we'd like to touch upon during the panel. And that is, next, species differences with respect to genetics, underlying genetics, epigenetics. While many of the genes are very similar across species, there are many that are not, right? And ontogeny, brain development also differ. How do we kind of compare what happens in the human brain development with that happens in rodents? And what kind of measure do we have to give us some accuracy or so confidence that we're really comparing apples and apples as opposed to apples and oranges? Behavior and systems biology, their behaviors are different. In terms of sleep cycling, for example, feeding behaviors, environmental impact of rearing conditions, and also epigenetic effects that affect humans as well. And then, of course, despite decades of research in the epilepsy field, we still don't really fully understand the intrinsic mechanisms responsible for seizure genesis and epileptogenesis. We're very good at describing seizures and actually sort of, both clinically as well as maybe experimentally. But do we really fundamentally understand what precipitates seizures or what actually terminates seizures, what the longer term impacts are? And then even more complicated than that are the comparative differences with respect to metabolism. And I think that we'll see that this is even a greater challenge in terms of applying this in the context of the epileptic brain. Next. So part of the discussion has to do with the practical issue. How do we facilitate meaningful, relevant translational research that will actually provide advances ultimately to the bedside? And here, I think the NIH has created a wealth of opportunities within its intramural structure to provide support and guidance for how to achieve some of these translational advances. And so the NINDS Division of Translational Research really is a fundamental partner in this and is designed to really accelerate basic research findings toward patient use for neurological disorders and stroke, epilepsy, by providing funding, expertise, and resources to the research community. Next. And if you take some time and go on the website, which is reflected below on the slide here, you'll see that there are a dozen or more mechanisms that are available for facilitating translational research, translational progress, and closing that divide. Next. So in this brief panel, we are going to have several experts weigh in on some of the questions that I've posed here. The first is Susan Masino, who's the Vernon Russo Professor of Applied Science at Trinity College, who does basic and translational research in the area of ketogenic diet treatments, and is also focusing on the role of the purinergic neurotransmission and adenosine as a potential mechanism for seizure control. Dr. Mary Ann Pelleymounter, who's basically the chief of the Division of Translational Research at NINDS, will provide great context and scientific perspectives regarding the questions. And she's very involved in many of the initiatives that are outlined in the NIH website. And the third panelist is Dr. Shai Silberberg, who is the Director of Research Quality at NINDS and runs the Division of Extramural Activities, and will also weigh in on this as well. So with that, I'd like to ask the panelists to turn on their video, and we can begin with some of the basic questions that we asked. So the first question really has to do with biomarkers. And I guess to the panelists, what are the fundamental questions in the ketogenic diet and metabolism-based field with respect to biomarkers do we need to tackle? You know, what are the key questions that have not been answered in the last 100 years? What is a relevant biomarker, and what should we be going after? I'm going to ask perhaps, Dr. Masino to maybe start off and provide some, context to that question. Yep.
Hi, thank you, Jong, and delighted to be here. I guess in terms of biomarkers, of course, we want something that's going to be predictive and reliable. And I think one of the issues that's been just a perennial problem is the diversity of epilepsies, and trying to find something that's going to be applicable to a condition that is so variable. A couple of the ways that I've approached it is kind of, from the bottom and the top at the same time. We've recently done a full metabolomic screen of cerebrospinal fluid, and also have tried to approach from patients, pediatric patients with epilepsy, and also trying to look at this from the animal research perspective as well. I can talk a little bit more about some details, but let me let the other panelists weigh in on this as well.
Yeah, Dr. Pelleymounter, do you have any thoughts on the biomarker question?
Yeah, I think one of the first things that you probably need to ask yourself is, what do you want to do with the biomarker? Because it really relates to how you would even go about finding the biomarker. So if you want a biomarker that gives you an idea of whether a therapeutic is engaged a target, then you want to get the biomarker has to be the most direct way to the mechanism of the therapeutic itself. But if you want to ask a question about how do you, let's say you're trying to, epilepsy is a fairly heterogeneous type of disease. So let's say you want to be able to sort epileptics into subsets for a clinical trial, and you want to make sure that that allows a more simple and direct design of the trial. Then, you may want to see what separates, what even behavioral phenotypic traits are separating those different categories, or what physiological traits. So I think one important thing, and that the FDA also suggests, is first figure out exactly what question you want to ask. This isn't my field, but I was reading a little bit about biomarkers, or just ways of measuring what the ketogenic diet does in the brain. And I've noticed there have been some interesting improvements, at least in the ability to do PET imaging and to ask questions, direct questions about ketone metabolism versus glucose metabolism in the brain. And how has that changed with the ketogenic diet and normals? And I think what isn't totally answered yet is, how has it changed in people with epilepsy or people with Alzheimer's disease, that kind of thing. So when you ask that question about biomarkers, it's so broad that I think you have to figure out, what are you in particular interested in knowing about? And I should just mention, our program at NINDS is focused on trying to get people to narrow down what that question actually is, and then validate it.
Yeah, that's an excellent point. And certainly, we've been using the term biomarkers for many years in many areas of medicine, and everyone's defined it very differently. And it wasn't until the panel got together to finally put some precision around that. And so it's a very important question for sure. One of the challenges in the epilepsy field outside of the ketogenic diet proper is that we don't really have biomarkers for epilepsy proper, right? And so while there are many correlative findings and sort of things that have been implicated, we don't really have things that are actually confident and believe are reflective true underlying biomarkers. So if we're attempting to define what we mean by a biomarker, and then looking at sort of bringing the basic science studies closer to the clinical studies and clinical information that we have, I think one of the key other questions that was asked, what is the limitation, or what are the limitations of trying to study or develop biomarkers when we're trying to compare, for example, rodents to humans? What are the pitfalls that we must be aware of in order to make comparisons? Because at the end of the day, as I mentioned in my introduction, there are fundamental differences at many different levels that we need to pay attention to. So here, I'd like to maybe have some panelists kind of weigh in on, what they view as being the major challenges for comparing mostly rodents to humans in terms of metabolism and metabolism-based treatments for epilepsy. So we'll start with maybe Dr. Masino again.
Sure. Well, one of the things that I found when I started working on the ketogenic diet in rodent models is that it's not very effective in a lot of our acute rodent models of epilepsy. And I've found the more chronic progressive models to be more successfully mediated by the ketogenic diet treatment. So it's kind of an odd situation where we have something that works in the clinic, but doesn't work in a lot of our animal models that we're hoping to be precipitating these biomarkers and predictions of efficacy and mechanisms of epilepsy, mechanisms of efficacy. So that's been, I think, one sort of slowly evolving realization, is making sure that you pick an animal model that gives you a robust response. One of the other ways that I've tried to do it now is expanding beyond just the rodent model. And I have a small study published actually in a couple of community-based dogs. And I'm just going to put it out there that I think dogs are an interesting model in that they have very diverse seizure types, and they share a lot of lifestyle characteristics. And there may be opportunities to develop more robust community-based dog programs where you can use genetics, you can collect all the lab results, and kind of start to even take what could be a complicated model, but maybe a good reflection of what's going on in humans.
Great. Dr. Pelleymounter.
So I think it's important to just keep track too of what is going to be the quickest way to answering your question, and what can you actually do at this particular time. So that leads me to the idea that if it is possible to measure things in humans, I just have found that, especially in the case of biomarkers, it's just the most efficient way to go is to just use human samples or human imaging if that's possible. But again, you can obviously be constrained by what you can do technically. In rodents, if you're just looking at a metabolic point of view, rodents are quite different than humans are. One of the things is just their size, and therefore their metabolic rate. They have non-shivering thermogenesis, so they use beta-3 receptors, whereas humans don't so much. Brown fat's an issue. Their diets are not really fat-ridden. So when you're trying to do a lot of things that are artificial with the rodent, and so then generalizing it to the human can be challenging. In a lot of cases, having to do with just adapting to things. I guess you could say generally rodents adapt similarly to a human, but the way they adapt, even some of the biochemical pathways that they use might be a little bit different than humans. So you just have to keep all of this in mind, and maybe even you might have to normalize sometimes to understand what's going to happen in the human or predicted.
So one of the questions that arises is, should we be investing in a continual fashion with more basic translational research in rodent models, or should we be pivoting resources more towards human biology, and so that taking advantage of some of the technologies that we have to study the brain structure and function at a more detailed level, or even the neurochemistry of the brain, for example.
Mm-hmm.
What are your thoughts on sort of where the field needs to go in order to make further progress? Maybe we can have Dr. Silberberg weigh in.
Oh, yeah.
Thank you. Yeah, as you know, as an outsider looking in, because this is not my field, I can't help but ask myself, what's being translated? What's there to translate? And I think there's a lot more fundamental, basic research needs to be done to come up with potential good pathways, mechanisms, and so on. We heard in the previous session. It's kind of all over the place, right? I mean, there are all these signals that this might be involved, that might be involved, but there's no clear mechanisms or pathways that one can zero in on right now and say, let's try and translate that. And then, of course, that brings back to the issue that you just discussed, that what would be the right animal model to study these potential translatable pathways. And I certainly don't have the answer to that, but I think that's something that must be kept in mind.
That's a good point. So one of the sort of things that one could, view the ketogenic diet and the metabolism-based treatment field as, it's grown considerably since the 1990s, but it's not a very large community. And if you looked at the panelists in the prior session, you can see that while many of them are doing very interesting, exciting, innovative work, there aren't a lot of researchers compared to other fields of medicine or even within epilepsy proper. So given the disparate nature of our research efforts, is there a way, sort of practically, mechanistically, or through the NIH, sort of kind of tackling that question of what do we need to do more fundamentally within the animal world to be able to translate to humans, as you point out, or do we sit there and sort of continue to explore, dozens, if not hundreds, of different potential mechanistic pathways and targets that will just amplify our underlying notion that this is a complicated beast we're trying to tackle, and we're never going to really fully understand kind of how distinct linkages are there. Is there a way of kind of honing in on sort of maybe, a team science approach, or convening a panel to identify maybe perhaps the best model, quote unquote, that could be used, and then having multiplicity approaches that have been taken and reflected by basic science researchers, whether they're looking at amino acids, or they're looking at the gut microbiome or other aspects, to sort of dissect this really apart. So what should we be doing in terms of trying to get that focus and ultimately that translation? And maybe we'll get back to Dr. Masino on that.
Yeah, so I appreciate that question, because I do think we still need to come at it from both directions with the team science approach. And in the, if we can do screening of human samples, I think that is an excellent approach, and then try and, work both from the top down and the bottom up. In the cerebrospinal fluid studies that we've been working on, what we ultimately came upon is that we're having to stratify the patients based on an optimal response versus no response, because the clinical, the gradient of clinical responsiveness is so all over the place, and you might be mixing, we found that getting a correlation between changes in different compounds and clinical response was not helpful. We ended up having to really just go to the extremes to try and pull out significant findings. So I think if we can use human samples to get really clear changes, and also work from the bottom up with animals, that might be our best approach. And we did find some compounds that were predictive of a good response, and some compounds that significantly went up or down in those two disparate groups. But if we didn't divide it like that, we wouldn't have found those clear outcomes.
Yes, thank you for that. There's also another challenge that happens on the clinical side. If you look at ketogenic diet centers throughout the world, and despite the presence of consensus statements by the so-called experts, many centers do things differently. Their protocols vary. And of course, the type of information, efficacy rates, the tolerability issues, all that can also vary. How do we sort of tackle that nut with regard to consistency of approach in order to understand these things and to study, human specimens, for example. Even with regard to genomics and metabolomics, there's huge variations there as well. It's much more complicated than giving an epilepsy patient a single drug or multiple drugs, because the diet is not a drug. It's probably arguably a multitude of different types of metabolic drugs that makes things much more complicated. How do we tackle that from the clinical side with regard to sort of precision, focus, and consistency of approach? Dr. Pelleymounter, do you have any thoughts on the clinical research side? What should be done or could be done?
Well, I think one thing that would go a long way, of course, is actually to know more about the mechanisms in the first place, which it's kind of a circular thing, right? I mean, you're always going to end up with a whole bunch of variable answers because you don't have one strong tool or even two, strong tools to work from. You don't have anything that's validated. So you end up, looking at, for instance, drugs that would cause insulin resistance, for instance, and also are epileptic drugs, or drugs that do not do that, and then look at how that separates clinical populations that way in terms of the responses. But I think in the end, it would be nice to be able to have sort of standardized approaches to look at things. But there's ways to do that, at least when you're collecting samples. But if you're actually running the trials, I don't actually know how you would do that.
So then, are you arguing that perhaps going back to Dr. Silberberg's comment that we need to maybe gain some further key insights, basic science mechanistically, in order to translate that to something that could be validated clinically, or and so how as sort of outsiders to the field and researchers in the ketogenic diet and metabolism-based treatments, what would you advise us as both basic translational scientists and clinical researchers to do in order to move the field forward and not to grow it in the same model of incremental, sort of growth, but something that actually will transform the field by generating a biomarker, by understanding key mechanisms that will enhance efficacy and improve tolerability, and all those things that we collectively would like to pursue together as a field? What would you advise? Dr. Silberberg?
Well, I don't like to be advising here. I mean, it's who am I to advise the field? But my feeling is I can't help myself, but but kind of thinking from an outside, looking in on the whole thing, and I ask myself, if there was no evidence that it actually works in humans, and particularly kids, which has been known now for almost 100 years, if we didn't have that, what evidence do we have that justifies translation or even going into the clinic? Okay, let's pretend we didn't know that it actually works. Where are we now that would justify starting a clinical trial? And I don't know the answer to that. My feeling is that there's not much, which would suggest to me that there's room to expand the basic research, make sure that any studies that we do in animals, will be well justified as far as the choice of the animal model, what is the question that's being asked. And then, of course, going into the clinic, you'd have to base it on that information, which we again have to ask, how much of it do we have right now? How good is it? How carefully was it done? As Dr. Masino showed in her talk, there are
1 minute remaining.
Conflicting results from all different animal studies on what works and what doesn't work and how it works. So I think there's a lot of soul searching, and I think that's what's so fantastic about this workshop here, which is an opportunity to think about all these things.
Yeah, thank you for that. Dr. Masino.
I just want to respond to what Dr. Silberberg just said. So I guess if we didn't know that it worked in kids, we would know now that, you know, metabolic pathology is associated with so many different conditions. And so that, I think, would be a rationale for trying to look at metabolism in general, and of epilepsy being kind of a rapid translation into something that we can measure in terms of brain function. So that's what I would say, what would be the rationale if we didn't know that it worked in kids. I think we have other pieces that kind of would support that, and also support looking into this as something that has implications beyond epilepsy.
Well, thank you for that. I think you know, we are at the end of our time in terms of the translational panel. And while we've tried to tackle some of these big questions, I think we're left with the feeling that we still need to define more crisply what we're trying to achieve here in terms of research goals. The translational divide still exists, despite, you know, decades of research. We haven't really begun to really crack the nut, and that's probably reflective of the complex epileptic brain, because that's also true, whether you're talking about biomarkers or trying to relate or translate findings in rodents to humans, and vice versa. You know, we have great challenges that lie there. So some food for thought, and maybe some you know, things that we could do in the future to align, focus better, and perhaps collectively move the field forward. All right. Thank you very much. And you know, this concludes the translational panel.